All right, folks, 18-8. 18-8 is the pancreas. All right. So, first of all, I want to remind you the pancreas has two functions, two main functions: an exocrine function. That means it secretes some products into ducts. This exocrine function is your digestive juices, like digestive enzymes and uh, bicarbonate, some digestive juice that neutralizes stomach acids. And then it also has endocrine functions. And what we're here to talk about in this particular part, this particular mini lecture, is the endocrine function of the pancreas. So the pancreas has endocrine functions, and it has these little islands of cells called islets of Langerhans. And it's the islets of Langerhans that secrete hormones. And there's different populations of cells in the islets of Langerhans. And we'll look at several of them. For example, the alpha cells secrete glucagon, the beta cells secrete insulin, the delta cells secrete somatostatin, I believe. So we'll look at these different islands of cells and what hormones they secrete.
The organs of the endocrine system secrete hormones directly into the bloodstream to assist in the regulation of various body functions. The endocrine gland discussed here is the pancreas. The pancreas is an elongated organ situated between the duodenum of the small intestine and the spleen, inferior and posterior to the stomach. It is both an exocrine and endocrine gland. The pancreas consists primarily of exocrine acinar cells and their associated ducts. Organized in glandular clusters, pancreatic acini secrete enzyme-rich pancreatic juice via the pancreatic ducts into the duodenum to aid chemical digestion. Scattered among the acini are small clusters of endocrine cells known as pancreatic islets, or islets of Langerhans. Each pancreatic islet contains 4 types of cells: alpha, beta, delta, and F cells that release hormones into the bloodstream. When blood glucose levels decline, alpha cells secrete glucagon, a hormone that stimulates the liver to break down stored glycogen and release glucose into the bloodstream. When blood levels of glucose and amino acids rise, such as after a meal, beta cells secrete insulin. Insulin stimulates cells to absorb glucose and amino acids from the blood and to store excess nutrients. Delta cells synthesize somatostatin. This hormone inhibits the release of insulin and glucagon and slows the activities of digestive organs, thereby slowing the entry of nutrients into the bloodstream. F cells secrete pancreatic polypeptide to suppress and regulate somatostatin secretion by delta cells. Together, these pancreatic hormones provide for the orderly uptake and processing of nutrients.
Okay, so there you go. Now, with the pancreatic cells, well, let's just revisit this. Make sure you memorize. Alpha cells secrete glucagon, beta cells secrete insulin, delta cells somatostatin, and F cells pancreatic polypeptide. All right, so make sure you get this. Uh, we're going to talk a lot about glucose metabolism right now. Now, remember, the delta cells secrete somatostatin that inhibit both beta and alpha. All right. What this does, and also inhibits your digestive mobility. So you have a big meal in your gut, and somatostatin is going to slow how it travels, how it is moved along, and this gives you time to manage the nutrients you're absorbing. Because now you're slowing how it moves from the stomach to the duodenum, from the duodenum to the jejunum, from the jejunum to the ileum. You're slowing this down. It allows you to it gives you more time to process these nutrients. All right. And then the F cells, the pancreatic polypeptide, inhibits delta cells. So all of this is going to be involved in really the delta cells and F cells are also involved in glucose control, because when you eat a meal, you're going to secrete insulin because you're absorbing glucose. It's going in your bloodstream, and that's going to cause the glucose to go into your blood cells. When you are in the fasting state, you're going to secrete glucagon, and you're going to break down glycogen into glucose. So all of these together are going to help control your absorption of nutrients. All right, this is showing you glucose homeostasis. Here's our normal level right here. I'm trying to get down to it. My pen's not working. Here it is right here. 70 to 110. Your glucose levels is normally 70 to 110 milligrams per deciliter. Remember, a deciliter is 100 mLs. Just in case you forgot what a deciliter was. If your glucose goes up, your beta cells secrete insulin. Insulin opens glucose channels in target cells. So now I have more glucose transporters in my target cells. So now the glucose enters my cells. All right. If the glucose enters my cells, blood glucose levels decrease and I come back to normal. All right. So there you go. Look at that. Look at this right here. This is important. If all of a sudden all my cells are, by the way, I shouldn't say all of my cells. Some cells don't need insulin to absorb glucose. They can absorb glucose without insulin. But many of your body cells need insulin before they absorb glucose. Well, if my cells are absorbing glucose, what does it do with it all? Converts it to glycogen. So it takes thousands of glucose molecules, hooks them end to end to end, and makes your glycogen. As you know, or maybe you don't know, but I'll just tell you, your liver and your skeletal muscles are rich in glycogen. They have lots of glycogen in them. Liver and skeletal muscles. All right. What happens if your blood glucose level decreases? Well, in between meals, in my post-absorptive state, let me just call it what chapter 26 is going to call it. Chapter 26 is, maybe it's 25, I forget, but one of the chapters you're going to do this semester, they talk about the absorptive state and the post-absorptive state. So in the absorptive state, that's when you're secreting insulin because you're absorbing glucose. So you have high glucose in your blood, high blood glucose. High blood glucose. So that's the absorptive state, and you secrete insulin. In the post-absorptive state, about 4 hours after your meal, you're in the post-absorptive state, and you have low blood glucose. And this leads to the secretion of glucagon, which breaks down the glycogen, which turns on mechanisms to break down the glycogen into glucose, to release it to the bloodstream to regulate your blood glucose levels. So if my blood glucose levels fall, I secrete insulin. Insulin leads to the breakdown in glycogen, where liver, skeletal muscle, that's a big place they're stored. And look at this as well. It also increases the breakdown of fat, so lipolysis. Because what happens there is, yeah, I'm breaking down glycogen and releasing glucose to my bloodstream. That doesn't matter very much if I'm going to burn the glucose that I'm forming. So if I start to burn fats, then I can conserve the glucose that I'm forming and allow that to keep my blood glucose levels in normal range. So I'll break down fats as well right here. And then I do this also. Glucagon also causes this. Increased synthesis of glucose. Now I'm going to tell you how cortisol does this. Cortisol is a big hormone that does this. This is called gluconeogenesis. So production of glucose from non-glucose sources is called gluconeogenesis. I don't typically even think of glucagon as being this huge gluconeogenic compound. It obviously is. Increased synthesis of glucose. That's called gluconeogenesis, production of glucose from non-glucose sources. New glucose production. Cortisol is a big one for this. Apparently glucagon is as well. I don't usually think about gluconeogenesis when I think of glucagon. I typically think of glycogenolysis. Glycogenolysis, the breakdown of glycogen. That's what I typically think about. But your book's telling you that glucagon can turn on gluconeogenesis, and that's fine. I'm sure it does. These are all hormones like glucagon and epinephrine and cortisol. They have either integrative or synergistic effects relative to glucose. So we'll talk about these in a few minutes. And there's some overlapping effects as well. This is the collage. This is showing you what happens when your blood glucose level goes up. You secrete insulin, that lowers your blood glucose level. When your blood glucose level goes down, you secrete glucagon, that raises your blood glucose level. And it tells you in there how it does both. All right. Diabetes is the disease of is an insulin disease basically. Now, is it directly insulin or indirectly insulin? Well, if it's type 1 diabetes, it's directly insulin. You have inadequate insulin production, and you typically take a shot of insulin a daily or more than one time a day sometimes. Type 2 diabetes is you might have plenty of insulin, but you don't have the insulin receptors, or somehow your cells are insulin resistant, and that's this right here, insulin resistant. So we can say you don't have enough insulin receptors, or for some other reason your cells are insulin resistant. You may make plenty of insulin, but your cells aren't responding correctly. We used to call type 1 juvenile, type 1 insulin-dependent diabetes mellitus, insulin-dependent diabetes mellitus. We used to call it the juvenile diabetes. We call type 2 non-insulin dependent diabetes mellitus. We used to call it adult onset. That's what we used to call it. We do know one thing, that obesity and type 2 diabetes are linked, are correlated. And actually losing weight is a good treatment for type 2 diabetes. So we do know that. All right. Now, all of these things right here can come back to one thing. They all can come back to one thing. What the heck is high glucose doing to me? Well, most reactions in your body are catalyzed by enzymes, but I can think of one that's not, and that is when you have glucose plus a protein, what you get is a glycosylated protein. Glucose adds to that protein, and it's non-enzymatic. The higher your blood glucose runs because you don't have insulin or you're insulin resistant, the more likely glucose is to attach to proteins. Proteins where? Proteins in your basement membranes. I mean, all your blood vessels have basement membranes to them. So if I'm running high blood glucoses all the time, I'm going to glycosylate the proteins in my basement membranes. Okay, what does that do? That thickens your basement membrane. What does that do? That eventually inhibits how much nutrient and oxygen can cross that basement membrane and get to your cells. So in essence, what you do is you decrease perfusion of nutrients to your cells because you've thickened your basement membrane. And it all goes back to this high glucose in your blood. So you get eye damage, and you get thickened basement membranes in your heart blood vessels, so you can have early heart attacks. You get peripheral nerve problems because you're not getting the nutrients to your neurons. You might have to have peripheral tissue damage right here. You might have to have your feet amputated. Why? Because you don't have enough oxygen to your tissues in your extremities, so the tissue dies and becomes necrotic, and you have to get an amputation. So on and on and on. And this is really just one horrible disease. And by the way, kidney degeneration. Why? Because I thickened the basement membrane of my glomerulus in my kidneys, and I no longer can filter correctly. I don't filter my blood correctly, so now the toxins stay into my blood. So you can see this is a horrible disease. And the key is to stay, as much as you can, euglycemic. Not hyperglycemic, not hypoglycemic, so not hyperglycemic, that's high glucose. Not hypoglycemic, that's low glucose, but euglycemic. And how to do that if you have diabetes is to do the finger stick and check your blood glucose periodically and take the appropriate units of insulin to keep your blood glucose level euglycemic as much as possible. This will keep you from having prolonged periods of high blood glucose, which will keep you from glycosylating your proteins and thickening your basement membranes and so on and so forth. Okay, so having said all of that, Because of this glucose glycosylating proteins non-enzymatic reaction, it also glycosylates a hemoglobin protein. And if I have a hemoglobin plus glucose, what I get is glycosylated hemoglobin. Well, hemoglobin is just a protein like any other protein. So if I have glucose in the presence of hemoglobin, I can have glycosylated hemoglobin. We call it another thing in the lab or in the hospital. We call it hemoglobin A1C. And hemoglobin A1C can be used because it just tells you where you're glycosylated, that's all. Doctors can measure hemoglobin A1C. And what it does is it gives them an average glucose concentration over 20 to 30 days, so say a month. So you really can't cheat your doctor. You really can't fool them. Because, yeah, you can eat properly right before you get your blood drawn, and you go in and get your blood drawn, and the doctor says, oh, you're doing great. Your blood glucose is 100. Keep up the good work. But really what you've been doing is eating gumdrops and all kinds of candy, and you're not supposed to. But you really can't fool him because he'll order and have the lab draw a hemoglobin A1C. And high hemoglobin A1Cs tell the doc that you on average have been running high, not just the day you had your blood drawn. Now he knows on average what you've been doing. So you really can't get away from that if the doctor chooses to run a hemoglobin A1C on you.
As blood glucose levels increase after a meal, non-diabetics release insulin. Insulin signals the cells in muscle and liver to take up glucose from the blood. This causes blood sugar levels to return to normal. In untreated type 1 diabetes, no insulin is released and blood sugar levels remain high. Type 1 diabetes is treated by injection of exogenous insulin. This insulin has the same effect as normal insulin. In type 2 diabetes, insulin is released in response to a blood glucose increase, but the cells do not receive the signal. One treatment for type 2 diabetics is Avandia, an oral thiazolidinedione that increases cell sensitivity to normal insulin. An effective treatment for obese patients is metformin, an oral medication that blocks liver synthesis of glucose.
So that metformin blocks gluconeogenesis. And when we get to the cortisol hormone, I'll talk more about gluconeogenesis. I already talked about it a little bit with glucagon, but I'll talk more about gluconeogenesis when I get to cortisol. All right, so that's type 1 and type 2 diabetes.